This protocol provides an accurate and reproducible method for quantifying 10 different lipid classes of Saccharomyces cerevisiae using a single extraction method and a single LCMS method. This method allows the detection and quantification of isobaric and isomeric lipid species having different chemical and physical properties.
For Saccharomyces cerevisiae culture, first add 5 milliliters of sterile 20% glucose stock solution to each of 2 Erlenmeyer flasks containing sterile Y and B medium to a final concentration of 2% glucose. Then use a sterile pipette to transfer a volume of an overnight yeast culture containing 5 times 10 to the 7th yeast cells into each flask and culture the flasks for at least 24 hours at 30 degrees Celsius with rotational shaking at 200 revolutions per minute. For lipid extraction, determine the total number of yeast cells per milliliter of culture and transfer 5 times 10 to the 7th cells into a 15 milliliter centrifuge tube. Add ice-cold nano-pure water to bring the total volume up to 15 milliliters and collect the cells by centrifugation. Resuspend the yeast pellet in 15 milliliters of ice-cold isotonic ammonium bicarbonate buffer for a second centrifugation and resuspend the pellet in 1.5 milliliters of fresh ice-cold ammonium bicarbonate solution. At the end of the centrifugation, store the pellet at minus 80 degrees Celsius until the extraction. To begin the lipid extraction, thaw the cell pellet on ice before resuspending the cells in 200 microliters of ice-cold nano-pure water.